actually most people don't seem to have a problem hearing me whether I have a microphone or not, to be honest. Especially if they're in the lab and they're not working. So hopefully, you've got that one.
Oh, sorry, I need to get it up.
Yeah.
Oh, we have to fill in for a minute.
Okay, right, well I'll carry on talking. So it's a really great pleasure for myself and Alan to come and see you all today. Alan is the person you're funding. I, as Jane said, have been looking into trying to grow these tumours for the last 3 to 4 years, and unfortunately there is no way of splitting your time. I wish I could, but sometimes when you're trying to do about 6 or 7 things at the same time, you just don't have the time to dedicate to one particular project that you really, really want to. And this is where Jane came in and said, you know, well, would it really help to have someone else working alongside you? Hence Alan. So Alan has come in, started work with us in November, and he's come in from a scientific background, but without the necessary experience that we needed in the lab. So he started to learn this up, and it has really helped me because it means that I can now concentrate on doing other things, some things that I really didn't need to do, Alan will be doing. So what I'm going to do today is give you an overview of where we are, why we're where we are, and what we're hoping to do in the future, and some of the real problems about doing this. So hopefully Jane will get the slides in a minute, otherwise I can talk through my slides.
Well, I get it to there, and I can't actually. Oh, there we are. It's a different presentation to.
Yep, that's it.
Okay.
That should be it. Yeah. So yeah, that's it. Great. So when we try to study cancers, there are various things you can do from a piece of tissue, such as the tissue I'll show you there. One of the things is you can take the tissue, like Newton in Bristol's doing, also with Ian Judson. You can take the samples that have been preserved as archival samples and study them. And that will give you a lot of information about the tumour. It will give you a snapshot of what's happening to that tumour at the point it came from the patient. The only problem with something like that is it can't really help you when you want to understand how you might get a response from your cells. So this is the question that Jane was wanting us to address. Could we grow the cells? So if we can grow the cells in culture, like these ones down here, then there's a chance to see what would happen if you put a drug on it. Would this happen? Would the cells stop growing? Would they change? Would they start growing even more? So that was the basic approaches that you can take when you're studying. And if you think about what's happening in cancer, a lot of people, when they have an overview of cancer, they think of something that's happening when it shouldn't be happening. And that's completely right. A lot of the time, your body is just doing something that it would normally do. It's just doing it in a way it shouldn't be doing it, or it's doing it more than it should be, or it's using things that it doesn't need to do. Some of the genes that are really quite important in cancer have been involved, we term as oncogenes, and that literally means a cancer gene. So these cancer genes, they're not just sat there to try and give us cancer. They're often got a particular role, and sometimes they're involved in fetal development. But they're only involved for a very small period during that time, and then they become switched on and behave longer than they should be, start doing things they shouldn't be. So understanding the processes is a combination of looking at the genetics and the behaviour. And a lot of these things that you can see up here, so growth suppressions, we don't get bigger and bigger and bigger. Our cells go to a certain point and then normally they would die off. And you get suppression of your growth. So one of the things that happens with cancer in the body is that's not happening. So we as individuals control our growth until we get to having a cancer development. And then the cancer will carry on growing when we don't want it to be growing. The only problem with this is when you take the cancer out and try to make it grow in a tissue culture flask, most of the time they don't grow, and this is one of the problems, and this is across the world. So when we're looking at any form of cancer, whether it's a breast cancer, whether it's a colon cancer, whether it's a lung cancer, or a GIST, there aren't many cell lines we can use. So this is one of the basic problems with doing things like this. So why would we want to look at the cancer? Well, as I say, we can look at the way it proliferates. So if we were to start the cells at day 1, you can see on day 2 how quickly they're growing in comparison. So these ones here on day 4 have quadrupled, whereas these ones here aren't doing much. What else can you do? Go back. Whoops. Go back. Initial start for a lot of drugs begins this way. So you'd have various cell lines, and these might all be, say, breast cancer cell lines, and you'd put cells, defined number of cells, into each well. And then if you can see the numbers, that's a number for a drug. So you put a low dose, and then a slightly higher dose, more doses, and a top dose. So as you can see, with all of these, starting with no drug, through to the most drug, the cell numbers and the cells that are growing are dropping off. So this would be a basic test. It might be a starting test that you'd look at. But for this, you might need to have 5 or 6 cell lines. So these are all from the same type of cancer. In fact, they're a thyroid cancer, which is also another fairly rare cancer. And you'd want to see whether it was working in one particular type or all of them. And it works on all of them. Even these ones down here, there's far less. How does this work in principle? I don't know if any of you have heard of PARP. It's a drug that's just come out and it's been licensed in America in the last year or so. It's coming across to England. It's going to be used for breast cancer. And it was the drug that introduced the idea of synthetic lethality. And essentially what that means is a tumour has a genetic defect that then means it's susceptible to a particular drug that has been tailored and designed to target that tumour. So essentially that's what we would like to be able to do. All the starting point for the work that identified PARP for the treatment of breast cancer, a lot of this was done in Sheffield by a colleague of mine called Helen Bryant, but she used some of these initial assays to do the initial work. Almost 20 years ago now this was done. And these are some of her, this is one of her slides I borrowed to try and put into context how sometimes these things take longer. Overall, as a scientific community, we're getting a lot better at doing these things. We've managed to collate more information, there's a lot more collaboration, so we can do things a lot quicker than we used to do before. But the timeline can still be quite long, especially if you're identifying a new drug that's never been used before. So the timeline from PARP, from the point of view of these initial assays, to going into treatment was almost 20 years. What else can you do? Well, using your tumour cells, you can artificially model the process of the tumour starting in one part of the body and then spreading to other sites. So for a tumour to do that, there are lots of membranes in the body it needs to get through. It needs to get into the blood vessels. It needs to get into the lymphatic. It needs to be able to move, not just sit there. So you can see if you put your tumour cells from your cell line that you've got into an assay where you've got a membrane in between with some artificial layers that mimic the structure of the body, you can see whether the cells would do it naturally and invade through these pores, or whether they need something to stimulate it. And I don't know, can you all see down there? This is Electron Micrograph, so it's a really high image version of these cells. So these are some tumour cells that have gone through these pores, and this is the bottom side, the underside. So these cells have gone through, they've pulled themselves through, a bit like they're crawling through this hole, and they're getting out the other side. And that's the sort of process they would do within the body. So if you set up an assay like this, you can see what factors are going to increase it, what factors are going to decrease it, and what you might want to target. Now, why is it important to have as many cell lines as possible? If you look around the room, none of you look the same.
Some better than others.
I make no comment. It's the same with cancers. No two cancers are exactly the same. So when you put cell lines together, you want to have a number to be able to give you an idea of what's happening. So in here, these are all from the same tumour. They're actually melanomas. And if you have a look, these are positive and negative responses for individual tumours against the same treatment that we've added in. And there's a real range of response. So using cell lines can help us manipulate and study what's happening. Having more of the same type gives us an even better idea of what's really going to work and what's not going to work. And this was the point that I was really we've been getting involved in doing this for a while as a part of a project that was funded by Sarcoma UK and the Sarcoma Trust in the first place. And as part of this project, we started working on sarcomas that had come from patients treated in Sheffield and trying to grow the cells. So these is an example of 6 sarcomas that we've now grown in Sheffield. These have been growing now for about 5 or 6 years. So they're growing really well.
Are they bone sarcomas?
These ones aren't bone sarcomas. We've got some from undifferentiated pleomorphic sarcomas. We've got a myxofibrosarcoma, some LMSs, and what we've now done, they've sort of got to the point where they're happily dividing within the space of 2 to 3 days. They might need to be subcultured. So they're growing pretty fast. So in fact, altogether, I think there's about 11 or 12 lines now from sarcomas, which actually has dramatically increased the number of sarcomas that are available worldwide. Some of these are the first examples of that particular subtype of sarcoma in the world. So we've already started to get a lot of requests from people all around the world to have examples of our cells so they can do their own research. And to that end, we've generated, this is, if you like, the information that we will provide with these cell lines that will be available from the university to researchers. And the researchers will then say what they want to do with the cell lines. We can make sure that they're not going to be doing anything profitable, that it's all for research purposes. They will be available for commercial companies, but the commercial companies, we are going to ask if they would give some money back to license it, which can come back to support our research on sarcoma. So it's all going to be generating more benefit and back to us. So with something like this, we show examples of what the cells look like for the original cell line, how fast it grows over a period of time, what its genetic profile is, what sort of changes we've got, and what else we've done with it, any staining we may have done, and so on. And that information will be available. So where are we now with the pores GISTs and GIST in general? So as Jane said, we sort of got involved in latter part of 2013, and we've had a number of 7 samples from patients that have had their tumours resected all over the country. And in all cases, we've managed to get cells to grow, which is brilliant. But when you look at a tumour, you have to also bear in mind that it's not just the tumour cells that are in there. There's often blood cells, there's also sometimes connective tissue, there's some normal cells, there's a real mixture. So when your cells first start growing, you've got a real mixture of cells in there, and sometimes, as I say, unfortunately, tumour cells don't want to grow, and we really don't know why that is always. So we keep the cells growing, and this is when it becomes very laborious, because you have to keep monitoring them on a sort of a fairly day-to-day basis. You need to judge accurately when you might be able to split the cells, divide them, and see if they can subculture them. And to put it into context, this is a cell line from a GIST, which has a mutated kit. It also expresses DOG. And as a control, we obtained this cell line from a place in Holland who sent us the cells over. So these are the only, to my knowledge, GIST cell line in the world that is available for research, and it is a mutated kit. So this is our positive control, because if we want to know what's happening and whether we've got the right cells, we need something to compare it against. So we're using this as a positive. We've also got some other tumours that also express KIT mutations and things like that. This one here we're going to use as our negative. It's our newest undifferentiated pleomorphic sarcoma. So this one's a relative baby. It's only been growing for the last year and a half. So it's fairly young in the culture. The ones at the bottom, can you see these? They've been going for 3 years. They've been going very, very slowly, but these are from a paused GIST patient. So on average, we might need to split these every 2 to 3 days because they're growing so fast. We might need to split this once every 2 to 3 weeks. I might need to split that once every 2 to 3 months. So that gives you an idea of why it's quite difficult to speed up some of this work. They're very slow growing. And all the time we're doing this, there's a risk that you might lose your cells to contamination. What are we going to do with these cells? I don't know if you can see, but some of the shapes on these cells is pretty much the same as we've got in the other sarcoma cells then. So we're hopeful that what we've got is what we want. So what are we going to take it forward? One of the ways we're going to do is we're going to look at the staining of the cells. So there are certain proteins that will be expressed in these cells that we can target and see how well they're stained and verify that. That's why we've got our control cell line. And you can look at these cells at low power or high power and work out whether they're actually expressing. So Newton Wong in Bristol is going to take some of our cells that Alan has got ready and prepared and do the staining. We've tried this in Sheffield. They haven't worked very well, and part of the reason may well be is proteins that are expressed by the tumour in the body because they need to produce that. Sometimes when you put it into an artificial environment, whatever protein is now being made available within that environment, so it's no longer expressed by the tumour. So it may be there's an issue with how we're preparing the cells, how they're growing now compared to how they're in the body. But all of this will hopefully resolve with Newton in Bristol. So that's one thing we're looking at. It's a slide is missing, I'm afraid, but it's all right, I can talk through it. So the other thing we're doing is looking at the mutations. So any cells that we get in, we want to be in a position to be able to test for all the mutations we know, whether it's SDH, whether it's the KIT, whether it's PDFR, all of these. So at the process at the moment, Alan's been setting up the PCR for these and running them through. So that's been sort of standardised. So as our cells come in, both the cells we've got growing it now, plus past samples that have been frozen down, and any future samples, we can test them in-house fairly quickly to see whether they're what we want or whether they're something completely different. And then finally, as Jane mentioned, there's Daniel Tennant, who's in Birmingham, isn't it?
Yeah.
And he suggested some various additives that we could maybe put into the media to increase. So that's something we want to try and do, to see, much as I showed you with those growth curves, whether you can increase the proliferation of these cells by adding things in. And then overall, hopefully, we're going to take this forward and be able to know with future samples how best to prepare and to increase our cell lines, with the ideally making several cell lines that can be used for research purposes, not just the one.